Does it seem that, okay, it seems that the microphone is on. So welcome to this course on computation analysis of single-cell RNA-seq. So my name is Martin Hemberg, and I'm a group leader at the Sanger Institute, just south of here. And helping me today are two members from my group, Tallulah Andrews and Vladimir Kiselev. And we're also hoping that Davis McCarthy, who is from the EBI, will be showing up here, but he will hopefully be coming. What is different from this time compared to the previous times we ran this course are two main things. The first is that we're going to be doing this over 2 days. And as you can see, you may have already seen all of the material that we'll be covering in this course is online, and it should be on your screens right now. We're hoping that we will be mainly done with the material sort of around lunchtime tomorrow, which means that in the afternoon we have some more open-ended exercises prepared that you can work on, but we also know from past experience that many people who come here already have a specific data set or a specific problem that they're interested in. So if you are one of those, then you should please make sure to bring that problem tomorrow, and we can basically try to help you work on that tomorrow afternoon. The other thing which is new is that we are recording this session to be put on the old internet for everyone to watch. So if you're not interested in being recorded, then you should not come up into this area here where I'm standing, because then you will be on movie forever. So I think that's what needs to be said. So yeah, so we're going to start by covering basically all the material which is here. So I've already gone through told you that what we'll be covering is on this web page. So, and we're hoping that this will be around for some time. So if you sort of feel that you don't have time to go over something, you can sort of look at it again later when you're home, and you can also send us emails if there's anything which is complicated and that doesn't seem to be making sense. On a technical note, in the next chapter are instructions for how to install everything that you need for this course. So you should be able to just run these commands on your own machine at home, and it should sort of install everything that you need to be able to work through the chapters. Right. I think that's it. Okay, so first of all, so we're assuming some background knowledge, prior knowledge of bulk RNA-seq. It's something that's been around for almost a decade now, and it's very, very widely used. It's sort of one of the most standard tools in transcriptomics and genomics and bioinformatics. And as you're all familiar with, I'm hoping is that it's a technology that allows you to measure the expression levels of different genes or transcripts across a population. Typically several millions of cells are used in each experiment. And so compared to the technology that it replaced, microarrays, it has several advantages in that it is sort of much more sensitive, and it is very useful for sort of comparative studies, because it does a very good job at measuring relative levels of expression. But it has some important shortcomings. Most importantly is that it takes large populations of cells, which we often know are heterogeneous. And so what you see is really the average of a large number of cells. You can't study heterogeneity, which is important, for example, during development, or if you're studying the brain, are typical, or immune systems are typical traditional examples of this. So a few years ago, single-cell RNA-seq became popular, and it is basically a technology that sort of advancement of the protocols that allows you to use the same principles as in bulk RNA-seq for very small amounts of RNAs. In fact, you can use RNA from just a single cell. So, and it was essentially at the end of 2013 when Nature Methods in an editorial said that a single-cell RNA-seq is now the method of the year. And it's around that time that it gained in popularity, because then the protocols had become refined and the sequencing cost had come down, so it made it a lot more accessible to many labs around the world. And the obvious advantage is that you can actually query the transcript levels of individual cells, so now we can actually understand heterogeneity much better. And there is a very large amount of technical development on the protocol side in this field. I would say that there are probably at least 10 different protocols that are sort of in current use to greater or lesser extent, and they have different advantages in terms of what they're doing. We are not going to go into great details on these different protocols in this course, but we have some links to papers that present them and compare them. What we are going to be talking about here is, of course, the computational analysis of the data and we're essentially going to be focusing on what happens once you get your set of reads back from your sequencing machine. That's the sort of the starting point for the analysis in this course. So here is a schematic which just shows the typical single-cell RNA-seq experiment. So that you extract your sample many times from a tissue, and then you need to dissociate the cells so that you can isolate them and look at them one by one. And it is sort of, again, my understanding is that this dissociation step is considered to be sort of one of the most critical ones in terms of ensuring the integrity of the sample and making sure that you have good quality, because it puts a lot of stress on the cells. So after that, after you've isolated your individual cells, you extract the RNA, you reverse transcribe it into cDNA, which you can then amplify and sequence. And once you have your sequencing reads, you try to form an expression matrix, which is the object that we'll be analyzing in the second half of the course. Right. So I've outlined here the sort of the steps that we'll be going through when we're doing the analysis. And the first parts here, the boxes marked in yellow, are steps which are sort of not specific to single-cell RNA-seq. These are things that you should do with any sequencing sample, any sequencing experiment. Whereas the boxes marked here in orange or brownish color are also important quality control steps, but here you need to use methods which are sort of specifically tailored for single-cell RNA-seq. And finally, down here in blue, we're listing some sort of more analysis or biologically related questions, and the sort of which one of these that you will be interested in depends really on what problem you're trying to solve or what your scientific question is. But these things up here you should always do regardless of what your problem is. Right. So what are the main challenges in analyzing single-cell RNA-seq data that are different from bulk RNA-seq data? Well, there are basically two main features of the data which differ. One is that because you start with such small amounts of RNA, you have to have a large degree of amplification, and anytime you do amplification, that can lead to biases. So we need to somehow correct or account for those. The other feature, which you'll notice eventually, in case you haven't already seen or heard of this, is so-called dropouts, which is a sort of prevalent feature of these data sets. And what happens is that you have, it manifests itself as having zeros in your expression matrix, which means that you have certain genes which, on average, have a relatively high expression level, but then you'll have a substantial fraction, say maybe 50% of your cells, where you actually have no detected transcription. And this is believed to be related to the fact that you're starting from very small amounts of RNA, and then you have relatively inefficient capture of the RNAs at the early stages before amplification. So what it means is that transcripts are dropping out before they can be detected. And from a mathematical point of view, this can be quite problematic because zeros are typically hard to deal with, as you probably know if you've tried to divide something by zero, or any normalization step will typically struggle with zeros. Okay, so another reason why it is more difficult to analyze single-cell RNA-seq compared to bulk RNA-seq has to do with distinguishing between technical variability and biological variability. If you're doing a bulk RNA-seq experiment, it's relatively straightforward, at least conceptually, to just split your sample into, say, 3 replicates, your same original biological sample. And then when you compare those 3 replicates, the assumption is that any variability that you see between them is due to technical factors. Now, for obvious reasons, this is much more challenging with single-cell RNA-seq, since it is possible to take one single cell, split the RNA into 2 pools, and then sequence them separately. But since we're already struggling, starting with low starting material, this is sort of not a viable option in terms of doing large-scale studies. So it's very hard to know to deconvolute the technical from the biological noise, but we're going to try our best in this course to see what we can do. There are 2 tools or methods that have been developed experimentally to help address these issues. One is spike-ins, which were introduced already for bulk RNA-seq. And the idea here is that you take synthetic RNA transcripts that you introduce at known concentrations into your sample, and then that gives you basically a fixed standard that allows you to normalize across different samples because you know exactly how much starting material that you have of each of these spike-ins. Then the second tool that is quite widely used is something called UMIs, or Unique Molecular Identifier. We'll be covering them in a later chapter, but the idea is basically that you add a short barcode to the end of each transcript, and before you carry out your PCR amplification. And what that means is that it will allow you to distinguish, so if you find the same transcript twice, by looking at the unique molecular identifier, you can tell whether or not it came from the same, if it originated from the same molecule. So if it was an amplification product, or if it actually comes from 2 different starting molecules. So this is quite useful for reducing the amplification noise. Okay, I think we're moving on to the next chapter, which is construction of the expression matrix. So what I forgot to tell you in the beginning is that the way we're going to be doing this is that we're going to be going through the material here. It's only now in the beginning where it's mostly me talking. In the later chapters, there are exercises interspersed. So what will happen is that we will be sort of telling you about this material, and then you will get to run some exercises before we move on to the next step. So very important is to do quality control. There's going to be a lot of that, and the main reason is that if you don't do QC, it's very easy to get artifacts or spurious signals in your experiment, which can often be much stronger than the actual biological signals. So removing anything that is of dubious quality is absolute key to getting a good, being able to do a proper analysis and getting good result out of your data analysis. So the first step is when you basically get your BAM or CRAM file back from your sequencing machine, and to check whether or not the reads are of good quality. So what most sequencers do is, in addition to telling you which base they thought you had at your read, they also give you a quality score assigned to that base. So you can analyze those quality scores using different tools. So two of them that we list here are FastQC and Kraken. So we've been using FastQC here, which is very easy to use. You download it, and then you can run it from the command line, as illustrated there. And here we're showing the output from FastQC. And as you can see here, we have the 125 base pair long read. So read position is shown here on the x-axis. And then we have the quality score here on the y-axis. And what you can see here, this is often the problem, that you can have slightly lower scores at the start and at the end of the read. In this case, we also have, for some reason, we have a dip in quality for two bases right here, somewhere in the middle of the read. We don't really know why this is for this specific data set, but it would be useful information for you as bioinformaticians to feed back to whomever carried out the experiment that there seems to have been this issue. In this case, we suspect that just having two bases of poor quality out of 125 is unlikely to be a major issue. So we're going to make a note of this, and then we're going to continue using this data set as is, and hope that it will not confound the mappers too much. All right. So the next step is that once you removed the poor reads or the bases at the start or the end of the reads that are consistently low quality, you can try to map them to a reference genome. So mapping to a genome is, of course, has been done for a long time, and there are several different tools available for that. We typically use an aligner called STAR. We find that it seems to have the best performance for single-cell RNA-seq data that we're looking at with the read lengths that we typically have. I think it's important to, if you doubt the quality of your aligner, you can obviously try a different one, and you can also conceivably try to change the parameters that you use for the aligners. What has happened in recent years, there have been new methods that have been published, so-called pseudo-aligners. The two most popular ones are called Callisto and Salmon. And what a pseudo-aligner does is that if you're only interested in looking at the transcriptome and not at the genome as a whole, you can improve your speed significantly by only aligning, by basically doing alignment and quantification in one step. So it has been shown that they are several orders of magnitude faster than the traditional methods, and they have comparable levels of accuracy. So if you have a very large data set, tens of thousands of cells, you definitely need to consider these methods, the pseudo-aligners. Whereas if you have a smaller data set, say only up to a few hundred cells, then you can probably get away with doing it the traditional way. So here we have just an example of how to use the STAR aligner. You also need to take into consideration what to do with your spike-ins if those are present in your data set. And essentially they need to be aligned to a specific, you need to add those sequences to your reference genome so that they can be aligned properly. And for UMIs, you need to remove the barcodes prior to alignment, because otherwise that can confuse the aligner. And what people typically do is that in order to make sure that you keep track of the information about the barcode, which is associated with each read, you can add this barcode sequence to the name of the read. Right. So the other thing I need to say is that it's not really possible to say exactly what is considered a good alignment. It depends on many different factors, and for example, how the library was prepared, and so on and so forth. In our experience, we typically see that for most mouse and human data, we get about 60 to 70% of the reads being aligned. Now, that is a sort of a rough guide. If you see something which is significantly higher or significantly lower than that, you should probably go and check and figure out why this might be the case, if there's some error going on here. Really what you need to do is important to consider is to look at, rather than trying to compare to some absolute reference, you need to compare sort of within the sample. You should expect that all the cells have roughly the same amount of percentage of aligned reads. So really what you're looking to do is to remove outliers, so removing cells that have significantly fewer aligned reads or significantly more aligned reads than the bulk of the population of cells. Okay, here we have an example. So this is a small data set where we have, I think it's 96 different cells here. And we have classified each read as either being uniquely mapped, mapped to multiple loci, or unmapped. And then we've sorted them based on the amount of reads. And as you can see here, we have the majority of cells, you have at least 2 million uniquely mapped reads, which is very good. What you can see here is that we have 3 genes here where we have fewer than 1 million uniquely mapped reads. So we're going to basically say that those are outliers, and we're going to exclude them from further analysis. You can also see here that we've sorted, as we sort, we have 2 cells where we for some reason have a few 100,000 fewer uniquely mapped reads than you would expect based on the sort of the typical trend here as a number of uniquely mapped reads tracks with the total number of reads. We don't really know why this is. We think it's useful to make a mental note of these 2 cells and see if there is sort of something suspicious happening to those cells further later on in the quality control procedure. But for now, we don't think that this small aberration is enough of a reason to exclude them from further analysis.
So the assumption is that you should have roughly the same number of map reads because we are working with cells that are coming from one tissue, right? So what if you are working with cells from 2 different tissues?
Yes, yeah, so that's a good question. So the question is that we're assuming here that we should have similar number of reads because we have the cells come from have a similar origin. So yes, it is true that if you sort of if you have cells from different origins, you may no longer expect to have this similar read depths. So that would obviously be something to take into consideration. If you think that you might have a bimodal distribution for sort of some biological reason, then that would obviously be useful to keep in mind. Right, so now that we've mapped the reads to the genome, we need to also check that we were able to capture the parts of the transcriptome that we're actually interested in. So for example, one issue that can often ruin an experiment is if you don't do proper depletion of the ribosomal RNAs. So then you can have most of your reads mapping to the ribosome, which is not very useful information. So what we can do is that we can use, again, standard tools, for example, RCQC, and investigate sort of certain key variables, as in sort of how many reads mapping to ribosomal RNAs or transfer RNAs, what fraction of uniquely mapping reads, and so on. So we have given you some examples of how to run these commands here. Again, it's very hard to give exact general guidelines for what to expect here. And again, you have to compare to the sample as a whole and look for outliers really and exclude them from further analysis. So one example here of a feature to look at is the coverage of the transcript. So what we have here is a sort of a metagene. So we've rescaled and renormalized the length of the gene to be between 0 and 100. And then we're just asking what fraction of reads fall into each part of this gene, this metagene. And as you can see here, as with many of these protocols, we have a 3' bias, so that we have very poor coverage at the start of the transcript, but significantly better coverage towards the 3' end. And here you can see that we have three of the, so each line here corresponds to one cell. So now you can see here that you have 3 cells that seem to be a little bit of an outlier here, in that you have, they behave differently from the overall population of cells. And so we like to take a relatively conservative approach to this. And so we decided to exclude these 3 cells from further analysis in this example.
What about that one that's waving?
So the question is about this wavily thing here. Well, we decided to keep it. It's if you decided to exclude it, sorry? We excluded it. There you go.
It's because it has very low number of reads, which is why you get all wavy noise.
Very good question. Okay, so now we can move on here. Right. So now that we know that we have good quality reads from our data set, and we also know that they seem to be mapping where we want them to map, and now we can start actually processing them to do some more interesting stuff. And what we typically want to do is that we want to measure, we want to aggregate reads across genes or transcripts. So for that, there are several different tools that were initially developed for bulk RNA-seq. For example, HT-seq or feature counts. And so what you need to give them is some sort of gene model for the organism that you're interested in, and then it will try to figure out what is the average expression level, what's the expression level of each of those genes. So here we're giving you an example of how to run feature counts. Right. So what is, of course, different from when you're using single-cell RNA-seq data and you have these unique molecular identifiers is that you can do an additional analysis which goes beyond what was possible to do with bulk RNA-seq data and what these tools are available to take care of. So in the next chapter, we're going to be talking about UMIs. So one sort of trade-off that you have to think about for your UMIs is that you only have a finite length of your reads, and in order to make sure that your reads map uniquely, you want to capture as many bases as possible of your transcript. But if you at the same time, since you're starting with hundreds of thousands of mRNAs in your cell, you would ideally like to be able to uniquely tag each one of them. So there is a trade-off between having sort of shorter UMIs, which will allow you to better map the reads, versus having longer UMIs, which results in poorer mapping, but at the same time it allows you to better resolve the individual molecules. And one of the reasons why it is important to one of the reasons why this can be problematic, and the reads may not be, you might think that if you have 125 base pair reads, and you only sacrifice 10 of those reads for your barcode, that should not be a big of an issue. The problem is that this barcode is typically attached after the poly A tail, so you'll need to have the entire poly A tail included in your read as well. So that will reduce the effective length. Okay, so I think if we scroll down here. So we mentioned mapping, removing the UMIs prior to doing the mapping, and now the question is, in principle, you should be able, these barcodes should be unique. In practice, they are not. And I think if we go down here, so here's what I was just discussing about the transcript. And often you'll have two barcodes. So you'll have one barcode that will allow you to identify which cell the transcript came from, and another one to identify which molecule it was. So now we need to count up the barcodes, and this is complicated for several reasons. So the first one is that you can have errors in your barcodes. So we have estimates that suggest that the error rate in these barcodes is around 10%. So what that means is that if you had an original molecule which was then amplified, so now you suddenly have 1,000 copies of it, if some of these at some stage during the amplification there was an error in the barcode, then it will suddenly look as if you have two different barcodes. So this will allow you to, if this happens and you don't account for it, you'll overestimate the number of transcripts that were present. Second problem is that you will have, again, a problem leading to overestimation, which has to do with problems with mapping, that you may not be able to resolve these uniquely. And finally, there is also reasons to suspect that you may have underestimates of the number of reads, because if you don't have a sufficient number of barcodes, because they're too short, to uniquely address your entire population of mRNAs for that specific gene. So, fortunately, there are ways of trying to address these questions. So there is a method which is not published yet, but it's available on GitHub, and I think they have a bioRxiv paper. Something called UMI tools, which tries to identify these types of PCR errors or sequencing errors in your population of barcodes. And then regarding the issue of the non-unique, not having enough barcodes, you can, people have tried to quantify this, and you can sort of basically set up a simple mathematical model to figure out when you have, what is the chance that you'll have this type of collision. And I think if we scroll down here a little bit, then what you can plot here is, so here's sort of data from an experiment where we have each dot here represents a gene, and the colors is a sort of false heat map to indicate the density of points, because there are so many points here. And then we have the number of different UMIs and the number of different reads here. As you can see, that they're roughly proportional, which is good, But if you look at the scales, you see that they differ by about number 5 here, which is in log 2 scale, of course, means 32. So that means that you have roughly a difference of a factor of 32 here between the number of reads and the number of UMIs. So I think we've reached our first exercise. Tallulah, do you want to introduce what this exercise, what we're asking them to do here? You should take the microphone.
Right, is this working? Yeah, so you should be able to, this data should be on your computers already. You should be able to open RStudio and copy that command in to load a data set. And then the exercise is just to, this is just an expression matrix of the number of UMI counts. I'm just asking you to try and correct the data for collisions and sequencing errors. So the collisions is the equation up here.